Number 1. Define genetics and its importance in fisheries. Genetics study of genes and heredity of how certain qualities or traits are passed from parents to offspring as a result of changes in DNA sequences. It is important to fisheries because most aquaculture species can be produce many offspring and large population with improved genetics can be breed quickly for improved production performance. The benefits of may include improved growth, resistance to disease, or robustness in diverse farming environment. Number 2 is, what is the chromosomal and chemical basis of heredity? According to the theory, chromosomal is a genes that are a unit of heredity and are found in the chromosomes. Chromosomal theory of inheritance came into existence of long after Mendelian genetics, and deoxyribonucleic acid is a chemical basis of heredity in organism. DNA is present inside the nucleus. It contains genetic information and instruction for protein synthesis. Number 3 is, explain the concept of population genetics. It is the study of genetics variation within among the population and the evolutionary factors that explain this variation. Its foundation is a Hardy-Weinberg's law, which is maintained as long as population size is large. Mating is at random and mutation, selection, and migration are negligible. Number 4. What is the importance of selection in fish stock management? The importance of selection in fish stock management is selectivity of rates both at the local scales, as in the direct interaction of individual fish with the fishing gear, contact selection, and at the stock wild scale population selection, as evidenced by the differential rates of fishing mortality at the age that are generally observed in stock management results. Number 5. Differentiate hybrids from inbreeds and plan a good crossbreeding program. Hybridization refers to the process of animals and plants breeding with an individual of other species or variety. But inbreeding refers to the breeding from closely related people or animals, especially over many generations. Number 6. Identify biotechnologies in fish genetics and determine how they could help increase productivity. Fish biotechnology are chromosomes manipulation and hormonal treatment which can be used to produce triploids, tetraploids, haploids, genogenetic, and androgenetic fish. This result in the production of individuals and lineages are IOPS, steriles, monosex, or highly endogamic fish. Culture fish are being improved for a multitude of traits, including growth rate, feed conservation efficiency, disease resistance, tolerance of low water quality, cold tolerance, body shape, dress out percentage, carcass quality, fish quality, fertility and production, and harvestability. Number 7 is, differentiate mitosis from meiosis. Mitosis is a type of cell division from somatic cells and for the asexual reproduction of unicellular eukaryotic cells. Meiosis is a type of cell division for the production of gametes in sexual reproduction. Number 8 is, differentiate DNA from RNA. RNA is a sugar ribose while DNA contains a slightly different sugar, oxyribose, a type of ribose that lack one oxygen atom. DNA is a double-stranded molecule that has a long chain of nucleotides. RNA is a single-strand molecule which has a shorter chain of nucleotide. DNA replicates on its own. It is self-replicating. RNA does not replicate on its own. Number 9. Discuss how and why DNA replicates. DNA replicate is the process by which a double-strand DNA molecule is copied to produce two identical DNA molecules. Replication is an essential process because whenever a cell divides, the two new daughter cells must contain the same genetic information or DNA as a parent cell. The opening of the double helix and separation of the DNA strand, the priming of the template strand, and the assembly of a new DNA segment. During separation, the two strands of the DNA double helix uncoil at a specific location called the origin. Number 10. Explain the importance of transcription and translation. The purpose of transcription is to make RNA copies of individual genes that the cells can use the biochemistry. The purpose of translation is synthesize protein which are the used for million of cellular function. The purpose of transcription is to change DNA into mRNA, and the purpose of translation is to produce protein from mRNA and tRNA. Transcription and translation take information in DNA and use it to produce protein. Transcription uses a strand of DNA as a template to build a molecule called RNA. Number 11. Discuss how protein amino acids are synthesized. Protein synthesis represents the major route of disposal of amino acid. Amino acids are activated by binding to specific molecules of transfer RNA and assembled by ribosomes into a sequence that has been specified by messenger RNA, which is has been transcribed by the DNA template. Number 12. Explain the principle of YY meal technology and the process on how to produce YY meal. YY meal produced by converting female into male population. Number 13. Explain the different kinds of artificial selection methods: mass selection, family selection, and multi-traits. Mass selection. The term individual selection and mass selection are often used interchangeably and they refer to selection solely based on the individual's phenotype. This well-known and widely used method of selection in animals breeding and for several aquaculture species can be used on characteristics that can be measured in the candidate's animal itself. The weight gain being most efficient in characteristic of high heritability. Family selection is a family referred to a selection method in which family groups are ranked according to the mean performance of each family and whole families are saved or discarded. The difficulty of the individual marking of the animals, which is still a fact in crustacean, led the recurrent use of this method is fish farming with a constant impediment of the emerge of the family effect. Multi-trait selection is the opposite of the previous method. The selection is based on the deviation of each animal from the average of your family. There is a choice of animals of the all family, then the method requires identification of the families. This may be achieved by maintaining them separate tanks, cage, half-as, and any other means of containment without necessarily tagging the fish. The pulsive growth rate in unreplicated half-as and any other form of containment fall into this category. Under such circumstances, selection between families would be misleading from the genetic viewpoint because of the compounding between genetics, units, and common environmental effect. If replacements are chosen so that every family contributes the same number of individuals to the next generation, choose one female and one male from each family. The effect population size is twice the actual. Number 14, advantage of crossbreeding. By using crossbreeding, we can combine breeds with a different strength and weaknesses to create an animal that is more balanced for relevant performance trait. Crossbreeding is not everyone. For example, some breed only accept purebred animal.